Hello everyone! Today I will be explaining about glycolysis. Glycolysis is one of the major stages in aerobic respiration that release energy to the cell. First of all, what is glycolysis? Glyco, which means sugar, and lysis means split. So, glycolysis is the splitting of sugar. Glycolysis process occurs in the cytoplasm of prokaryotes and eukaryotes. There are two phases in glycolysis process. The first one is energy investment phase and the second one is energy payoff phase. When a person eats foods that contain carbohydrate, glucose will enter the cell and receives a phosphate group from ATP. The enzyme hexokinase is responsible for the transferring phosphate group from ATP to glucose. The role of ATP here provides the phosphate group as well as the energy required to attach the phosphate group to glucose. ATP is hydrolyzed to ADP and energy is released at this stage. This stage is called phosphorylation. Glucose now become glucose 6-phosphate. Glucose 6-phosphate is rearranged to form its isomer, fructose 6-phosphate. This reaction is catalyzed by the enzyme phosphoglucoisomerase. Another ATP is invested in this stage. The enzyme phosphofructokinase transfers the phosphate group from ATP to fructose 6-phosphate to form fructose 1,6-bisphosphate. Now the sugar is ready to be cleaved. Fructose 1,6-bisphosphate is split into two 3-carbon sugars, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These two sugars are isomers of each other. This is the reaction from which glycolysis gets its name. This reaction is catalyzed by the enzyme aldolase. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are isomers and are interconvertible. This process is called isomerization, which is catalyzed by the enzyme triose phosphate isomerase. That is the end of the energy investment phase, where ATP is used. Now let's begin the energy payoff phase. Glyceraldehyde 3-phosphate undergoes dehydrogenation to form phosphoglycerate. Dehydrogenation is a process of removal of hydrogen. NAD+ in the cytoplasm is the hydrogen acceptor. Thus, it receives hydrogen and forms NADH. This reaction is catalyzed by the enzyme phosphoglyceraldehyde dehydrogenase. Next, the phosphoglycerate reacts with inorganic phosphate to yield glycerate 1,3-bisphosphate. This process is called phosphorylation. ADP is phosphorylated to form ATP. The phosphate group is removed from glycerate 1,3-bisphosphate to form glycerate 3-phosphate. This process is catalyzed by phosphoglycerokinase. Glycerate 3-phosphate rearranged to form glycerate 2-phosphate. The phosphate group is shifted. This reaction is catalyzed by phosphoglyceromutase. Of water is removed from glycerate 2-phosphate, which results in the formation of a double bond. The product formed is phosphoenolpyruvate. This reaction is catalyzed by the enzyme enolase. Lastly, phosphoenolpyruvate transfer a phosphate group to ADP to form ATP, and pyruvate is formed. The enzyme pyruvate kinase catalyzes this process of substrate-level phosphorylation. Pyruvate form then enters the link reaction. That's the end of the energy payoff phase. As you can see from the previous process, ATP or energy is used during the energy investment phase, while in the energy payoff phase, ATP and NADH is produced. Glycolysis is the source of ATP and NADH and prepares the organic molecules to be further oxidized in the Krebs cycle. One molecule of glucose yields 2 ATP, 2 NADH, and 2 pyruvate molecules through the glycolysis process. So, that is how ATP or energy is yielded from the glycolysis process.